on the QR code, and you can submit the questions and also vote for the questions, so we can also select the questions for the Q&A at the end. Let's start. Today's agenda is on Acies Bio presentation on how we can use the strain optimization approaches and which strain optimization approaches we are using, both the high throughput strain optimization and ALE. Today we will be presenting myself. I'm Martin Kauszczyk, Head of Business Development. Then we have Gregor Kosec, Dusan Goranovic, and Helena Joshevets, who will be tackling more technical parts of the discussion. Firstly, a little bit about Acies Bio, what we are and what we are doing. At Acies Bio, we are developing new microbial technologies using our Smart Route platform, which is basically a streamlined development process for microbial technologies, where we can start really with an idea, do the starting strain selection and pathway design, and then throughout the optimization steps of both genetics and non-GM based technologies, optimize the strains first in a really small scale using high throughput screening of microfluidics, microAL, and then through further strain optimization and evolution go towards more industrially relevant environments, iterate these cycles, and when the strain is sufficient enough, we go into the bioprocess and DSP optimization first on the small scale, and then with the developed bioprocess and DSP, we scale the production process in our commercial demonstration facility with up to 10,000 liters of fermentation volume, and also do the contract manufacturing. And we are expanding the contract manufacturing facility to 30,000 liters in the next year. So stay tuned for the news regarding that part of our development. But for today's talk, we will be mainly discussing these first parts of strain development and especially optimization and evolution. One of the key parts of our technology offers are also our technology platforms. Those are the strains, genetic tools, screening protocols, and bioprocess solutions for production of diverse sets of biological molecules such as lipids, functional proteins, terpenes, secondary metabolites, and many more. And those can be used to speed up especially first proof of concept and first parts of the development of the technology, to basically de-risk the technology early in the start of the development. We've done that for basically 19 years up to now, working with industrial partners from different parts of the world, more than 90 of them in food, feed, ag, bio, industrial, and pharma segments. We've come to develop projects from all different stages, from really scratch discovery projects. Many times we enable the technology to come, that your ideas come to a pilot scale, and then in the last part, we scale that technologies to industrial scale, either for big production, but also small scale production projects for more specialized molecules. And we've done that for diverse industry segments, such as specialty ingredients for food and feed, pharmaceuticals or nutraceuticals, industrial chemicals, and agriculture. And because of that broadness in the industry segments that we work for, we are also using tailored business models, from milestone-based development of your ideas to co-creation projects where we invest also our strengths and our know-how for development of novel joint products and joint ventures. And lastly, also produce the material either as a scale-up campaign or white label CMO production for the technologies that we developed. So that's Acies Bio in a nutshell, and we will be focusing today, as I said before, on really the key aspects of how to develop and optimize the strains. And I'm inviting Gregor Kosets, R&D Director at Acies Bio, to present our view of different strain optimization approaches that we have.
Thank you, Martin, and welcome everyone. Let's start. So where do these evolutionary or non-GMO methods actually come from? So a long time ago, our ancestors looked at the field, or at their fields, and noticed that among the thousands of plants growing there was one or some which were larger, had more grains, or had larger grains. And instead of putting them in the bread, they stored those grains and used them for the next year's sowing of the field. And through iterations, so through the millennia, and using this process, we got to the plants and to the crops that we are mainly using as food today. So that was the start. And then another big step for these methods was the invention or the discovery of antibiotics. So there was quite a long time between Fleming's discovery of the fungus which produced penicillin and the actual application and usage of the antibiotic in the clinical setting. And one of the main drawbacks was actually that the fungus produced the antibiotic in very, very low concentrations. And therefore, in order to assure supply for the global demand, which was huge, of course, they had to find strains which produced more. And they looked for that by observing colonies which had larger inhibition zones than their parents. And in this way, again, through iterations, they found a better strains. And this procedure was then used by many pharmaceutical companies throughout the 20th century in order to develop new antibiotics. And this procedure was actually the foundation of their technical and also economic success. But then, of course, 50 years ago, we started using genetic engineering as a much more targeted and much more quicker way to engineer microbial strains. So the question is, why do these evolutionary methods still matter today? And we'll have a look at this in today's seminar. There are many aspects why this is still really important. Yeah? The first aspect is, of course, the regulatory acceptance. So for some markets, for example, in food and agriculture or in environmental applications, people still prefer to use non-GMO development methods, either because they are favored by regulatory agencies or because they're favored by the final customers and all of us as consumers. And that even more importantly, these methods, evolutionary methods, are really complementary to genetic engineering approaches, which we all use today in most of our developments in biotechnology. And the reason for that is that evolution is an unavoidable principle of engineering biology. So either we like it or not, evolution will happen. And the idea is to use it in our favor, and of course not allow it to maybe damage the strain that we have created. And therefore, it's really important in order to have efficient bioprocesses and robust bioprocesses to control to control for any evolutionary processes that might be going on. So let's continue with the two main engineering approaches that we'll have a more detailed look in today's webinar. So there are two main principles. One is adaptive laboratory evolution. So this consists of prolonged cultivation of bacterial or microbial cultures in selective environments. So due to this selective pressure, mutants are generated which have increased fitness in such environment, for example, in the presence of an inhibitory compound. And of course, in order to use this principle and this approach, the trait of interest, so what we want to achieve, has to be correlated very closely with the fitness in selective environments. So the strains which grow better must actually correlate with what we would like to achieve. Yeah. The other principle that's called directed evolution, or sometimes even classical mutagenesis and selection, is slightly different. So this consists of iterative rounds of genetic diversification, most often some sort of mutagenesis, and then screening of the mutants that we obtain. And finally, after we screen and select the best ones, we amplify them and use them as parents for the next generation. So here, this is usually focused really on a specific target molecule, which we are trying to produce with the microbial process. And of course, it's based, one, on high throughput cultivation, and of course, a very good assay, which also has to be done in high throughput. Now, how do these methods actually fit in Acies Bio SmartRoute workflow? Clearly, they are carried out in the context or in parallel with the synthetic biology-based methodology in the initial stage of the development, so in the strain engineering part. So in the classical synbio circle, we have the design, build, test, and learn phases. And of course, this is again an iterative process. In order to do that in high enough throughput, we usually use microtiter plates, which you see in this figure. But of course, the process can also be done in different scales. Now, going specifically to the evolutionary methods, the circle is actually a little bit shrinked quite often. When we really focus on these methods, we quite often only have two phases. So it's only the build and test, correlating with the iterative circle I explained before. Yeah? So you basically mutagenize the culture, get single colonies, screen them, get a new parent, amplify it, and use that for the new round of mutagenesis process. On the other hand, very often the circle is actually expanded. So sometimes, in addition to the testing phase on the shaker scale, we also validate the strains in bioreactor scale, which is really important in order to assure the correlations between what we are doing in both scales. And in such an expanded circle, the learning phase really becomes also of key importance. So you would say, in the usual synbio, of course, you usually introduce a strain or a metabolic pathway, and the learning phase is basically learning whether the gene actually did what you thought or hypothesized to do. So, and you could say here, so if we just do the mutagenesis, what do we actually learn in this cycle? Yeah. So the first thing, of course, that you learn after doing a cycle, you learn which non-GMO approach actually works best for a particular situation. Yeah, so which mutagens, maybe which metabolic inhibitors, and so on. Then what you also learn is whether you have correlation between the high throughput assays and bioreactor performance. And if you don't, of course, you have to mitigate that quite soon. Yeah. And finally, using omics techniques, mainly genomics and transcriptomics, you may realize or find out which previously unsuspected genes and metabolic pathways might affect products formation, which of course then gives you new ideas of what you want to do in a more classical synbio way. Again, it's really important to stress. So the analytical capabilities are of key importance here. At Acies Bio, we understood that quite early, so we have done a lot of work in order to have the optimal assays available and also optimal chromatographic devices. Yeah. So basically, when you think about the analytics, throughput is of key importance. That's why colorimetric and fluorimetric assays are really favored when it's possible to use them. So they have really, fastest times and also highest throughput. However, most of the time, we actually rely on the rapid HPLC analysis. So the methodology here is quite specific. You need to have methods which are extremely robust and have short running times. Of course, when usual HPLC detectors are not enough or the concentrations are too small, we use LCMS and GCMS machinery, which allows us to basically work on almost any kind of molecule which can be produced by microbes. And really to stress out, good analytics really enables powerful non-GMO methodology. I'd also like to touch, again, the complementarity of synbio and evolutionary approaches. Why is it so important to combine them when you do this kind of development? So on this graph, you can see a situation where we transform the parent strain with a single genetic construct. And then we analyzed 112 transformants for production of the desired compound. And you can see that the differences in productivity are actually huge. We have several folds. So some of them are really, really good. Yeah. Most of them are in the middle, and some of them are really, really bad. Yeah. And this really means that you have, even if you do just genetic engineering, you have to screen enough candidates in order to find the best ones. And why is that? Yeah. What is, really, when we look at this design, build, test, and learn cycle, yeah, how does biology here differentiate from other engineering disciplines? Yeah, you can imagine this cycle on an example. If you want to fold a paper airplane, so basically you try different designs, you build these small airplanes, and you test them by throwing them in the air and looking which one will fly furthest. And of course, then you learn and combine your learnings and make a new generation of models and start again. Yeah. However, when you engineer a strain, a microbial strain, the situation is fundamentally different. And why is that? Because the strain had its evolutionary past, which led to the parent that you used. Then, of course, you carried out the transformation of genetic elements, but you have to realize that after that, the cells started to divide and grow. And when they start to divide and grow, they experience different evolutionary futures. So that they might change further on, and you have to, of course, select the ones which change in the way that is favorable to what you want to do and not in the way that is not favorable. And you also have to realize that when you later on inoculate these strains in the bioreactor, the evolution will continue to happen. So at the end of the bioprocess, you will not have the same culture that you had in the beginning. So clearly, what can you do in order to control this process and lead it in your favor? Yeah, clearly the first step is really screening a large number of transformants and analyze them for highest product titers. Maybe in the best ones, some evolutionary changes already happened and will be maintained in all further work that you do. So the other really important aspect is assessing the stability of these engineered strains. So here it's crucial that once you run a bioreactor, if the strain is unstable, it will probably start evolving in a way that will throw out the genetic elements that you put in. And this is really important nowadays, when people feel that economics of biotechnology will be greatly improved through continuous bioprocesses. And you can imagine continuous bioprocesses are extremely lengthy, and the cells are all the time exposed to high concentrations of the product. So clearly, what is happening in such processes has to be carefully monitored in order to achieve the goals. So really not conclude, but open the discussion then for Dusan and Helena. At Acies Bio, we favor combining the genetic engineering and evolutionary engineering approaches. And this combination is one of the key pillars of the smart root approach. So basically, rational design really sets the direction, but then the evolutionary engineering fine-tunes and adapts the microbe for realistic conditions. And basically, this gives you access to broader solution space. It gives you, most importantly, robust strains, which are then scalable to larger volumes. And I can't emphasize enough the understanding of strain stability in different conditions. And then at the end, the regulatory hurdles which come when you want to, of course, register the product might be lower if you used quite a lot of non-GMO methodology.
Okay, thank you, Gregor, for this insights into how we are tackling the development using different approaches. I think it's very insightful to see that it's actually a combination that is of different methods that many times give much better result and much quicker result. Let's see how we actually do it. So what are the key features of the high throughput strain optimization and adaptive laboratory evolution? I'm inviting Dusan and Helena to take over, and just keep in mind that there is a Menti open for questions. So if you have any questions, just feel free to write it in, and we'll answer it in the end.
Okay, hello everybody. So I will explain a little bit more about how we actually do high throughput screening at shaker scale and what are the key steps that ensure our success. So high throughput screening is usually done, using these miniaturized assays at shaker scale where we test large microbial libraries, and this is done at least in thousands, if not tens of thousands, depending on the throughput. So the screening itself can be completely random. So this is when we know nothing about the strain, no background knowledge, no knowledge of biosynthesis, and so on. And as Gregor mentioned, usually this means a bit of learning in the beginning, and then the further we go, the more we know, and we can draw some conclusions. And then this approach can become more like semi-rational, where we can already rely on some existing knowledge and draw some parallels between the actual phenotype changes and mutagenesis that we introduce. So usually the targets are quite clear for everybody. I think usually this is every higher titer, better space-time yield is usually the most industrially relevant. But also we have quite a lot of experience with improvement of byproduct reduction. So this is more related to the actual product, or undesired metabolites, which affect the overall process in any ways, most often DSP. And also when the product itself is biomass, or the product is within biomass, then growth rate and overall amount of biomass is also a target for improvement. So the evolutionary part, of course, is very slow. It happens in nature, but yeah, our job is to really speed it up so it becomes feasible to be done in a relatively short time. For this, we introduce genetic variability in populations using mutagenesis. We use different chemical and physical mutagens, and of course, even this needs to be controlled at some point because the idea is to introduce the minimal amount of mutations to enable, statistically reachable aims to identify good mutants, but then again, not to be too harsh on the cells because you can also cause unwanted deleterious mutations, which are then eventually start resulting in the slow loss of positive traits. So for this, this is a typical graph which shows correlation between the kill rate, which happens when you expose the cells to a mutagen, and then how we try to find the optimal exposure to have a good frequency of mutations, but not overdo it, to say it like that. So that's how we use several phenotypic, let's call them markers, which tell us which dose to use, either concentration or time of exposure to a certain mutagen to reach the desired effect. Once we have this library of genetically variable cells, we of course need to screen them. And here is where the actual throughput comes in. So we like to help ourselves when possible using pre-screening assays. These are, consider them like a sieve, which basically focuses or concentrates our effort to test already enriched populations and not do it like completely randomly, because of course then you also screen for the lower producers or the average producers, which are not interesting. So we mostly use, I would say, two pre-screening approaches. One is microfluidics, which Helena will expand on a bit later. And we also do like semi-quantitative assays. Usually this is done on agar plates or disk assays. So these are quite, cheap to do. They enable a really good high throughput, and they give you a good, let's say, a sieve, which these candidates then go on to the main screening. The main screening is already done in as closely relevant conditions as possible. So this is a relevant industrial media, but of course everything happens in small volumes, most often in deep well plates, which enable several thousand per experiment, depending again on the type of organisms, which is quite of course different. So if these are nice simple bacteria or yeast to work with, single cell growth, those are the easiest. But we also deal with quite more complex organisms like filamentous fungi or actinomyces. These usually have a bit more tricky to handle. So for this, then you have to balance the throughput with the volumes you use. Then later on in flasks, you're getting there to more relevant conditions. Again, larger volumes. This is more for retesting. And most importantly, all of this needs to be also demonstrated bioreactor scale, because that's where it matters. And of course, if you see good correlation between shaker and bioreactors, we know we're doing good. So we just continue doing it the same way. If these correlations are not well visible, then we know we need to change something in our assay to achieve relevant results, which then result in successful scale-up. And since I mentioned microfluidics, I give my word to Helena to expand a bit more on that.
Okay, so maybe I tell you a little bit about ultra high throughput methods that we use here in Acies Bio. So we actually use two of them. One is FACS, is fluorescent activated cell sorting, which is an old method which you probably all know about. So this is the technique when you sort the cells based on fluorescence. It is usually used for screening of intracellular products or sorting based on cell size, which means that actually it is mostly used for yeast cells here in Acies Bio. But here we also developed our own smart fluidics workflow where we based the technology on fluorescent activated droplet sorting, which means that it can be used also for bacterial cells and extracellular products. So in both cases, you need to have some kind of fluorescent readout and good correlation between your trait that you're searching for and the fluorescence. The easiest way is if your product is already fluorescent, but in some cases you can translate your trait into the fluorescence with an assay, or maybe some intermediate product is fluorescent, or you add text to the product. So it actually, the methodology, it is useful if you are searching for some rare events that happen really rarely, or if you want to discard the bad performers really early and you don't want them in your larger scale screening. What is also really important for this smart fluidics workflow is the conditions of cultivation of the droplets to be as close to the larger scale as possible to make the selection really right. So maybe I explain a little bit about the smart fluidics workflow. You start with the strain library. It can be from mutagenesis, as Dusan already mentioned, but it can be also some kind of environmental sample, or it can be also from the library from the mutagenesis. So you want a really large pool of strains with, and they have to be really diverse. So then it is really crucial to dilute this library enough to get one cell per droplet. So a lot of droplets is actually empty, and some of them have one cell in it. And then you cultivate them in relevant conditions to get growth and expression of the product, and then you screen them for fluorescence. And here on the chart, you see this kind of fluorescent readout. It is similar to the FACS readout. And then you set up a sorting gate. It's usually the highest fluorescent strains that you're searching for. And then these selected droplets or strains go into the separate channel where you collect them and screen them on more detailedly in a larger scale. So here is one showcase from Smart Fluidics. Here we have VB2 producers, a strain library of VB2 producers that were mixed in a strain library. And we did, of course, encapsulation steps and then cultivation. And then we sorted different fluorescent strains into separate tubes. And then we screened them also on a larger scale. And what we saw that this correlation between the high fluorescence and good performance on larger scale was really good in this case. We got really good performers with the high sort, high fluorescence sort, but with medium fluorescence sort, we got really diverse strains. They were probably poorly growers and less stable than these really high fluorescence strains. With low fluorescence sort, we got bad producers, which is also was shown on shaker scale. The strains that didn't produce vitamin B2 were not even seen on this fluorescent chart. Now I give my word back to Dusan.
Okay, thank you. So to summarize this high throughput part, let me just emphasize what are again the key parameters here which are important for it to work. So the assay of the design of the assay, of course, is very, very important because you must be in relevant conditions to then enable and maximize the possibility that this will, of course, scale up. So this is the saying, you get what you screen for. And if you don't set up your assay well, it will most often mislead you and you will choose the wrong strains. Also, sensitivity of the actual assay is important because this kind of dictates how much of a difference you will be able to detect. Sometimes the technology is already really well developed and you're not looking for several factors of improvement, but maybe only 10%. And of course, this means that your assay needs to be able to see this. So you need a good resolution where, again, very good analytics is important. Of course, shaker scale has its limitations, deep well plates, especially because these are small volumes. So again, you have to design your assay so to reduce the negative effect of these limitations, which cannot be completely avoided, again, to prevent the wrong evolution to occur and you picking the wrong candidates. Analytical throughput was already mentioned. Of course, this is one of the possible bottlenecks when you really want to do a lot of samples in a short time. You have to develop the methods to have better throughput, and there is a fine balance between quality of data and throughput, and this, of course, needs to be really checked in the setup stage when you prepare for this work. Since many of the organisms are quite tricky, so the easy ones, again, much easier to work with, and the more difficult, slow-growing filamentous organisms, those are also time and more labor demanding, so usually this progress is a little bit slower, but again, everything can be adapted to make it work also. False positives are a reality of any assay, and this is it's okay. And of course, it's important to separate the actually good ones from the false ones. And here is where the retesting comes in. You have to do several iterations to demonstrate the strain stability and reproducibility. And of course, really use good controls to know what you're doing, basically. Scalability yes, probably worth mentioning again. This is the most important thing. So all the candidates identified at small scale need to be checked in bioreactors as soon as possible. So that's why we usually do relatively small rounds and prefer to do more of them. So we don't really invest too much time only on shaker work, where you basically have to work blind, and then maybe after a very long time you figure out that this is not scalable. So we try to get to the reactors as soon as possible, and once again we see this good correlation, then we know we can rely on our assays. So just to conclude, to give you some examples, this is a good example of how many different traits you can actually affect using non-GM approaches. Here we had a Bacillus strain, which was going to be used for our bioapplications, and the aim was to improve several traits. Initially, there was an undesired metabolite, which also had antibiotic activity, so this was not something they wanted to have. So first we had to find a non-producing strain, and using these quick pre-screening assays, this was very, very effective, and we got a few candidates already in a very short time. The next step was to improve the productivity of the actual target product, which was an antifungal compound. So again, using several iterations of strain development, we really got significant improvements, several factors of activity. Then came more like bioprocess development work. So we do this in parallel, of course, which is very important. So we develop media and bioprocesses, and we managed to design a completely defined media, which ultimately was even better than the starting complex media, which gives you even more control of the bioprocess and has a really easier DSP. Since this was a live product where live cells are present, we maximized the biomass as well as sporulation of this Bacillus strain, because ultimately we want to have good survivability. And another thing that was a problem during actual production was that the strain was producing some biopolymer and the broth was very viscous. So we needed to fix this to be able to manage it. So we found a non-viscous mutant strain, again using a non-GM approach with the pre-screening. And this ultimately resulted in a much, much more efficient technology compared to where it was started. Another example, which I think is one of the most difficult things when you do classical strain improvement, and this is starting with already really high titers. So this can either be already engineered strain, which went through numerous rounds of mutagenesis, or it can also be a genetically modified strain, which also already has really everything is really high and efficient and expressed and overall, so you have really high industrial titers, and then they still want you to, squeeze some more out of this strain. This is where the accuracy of the assay comes in. So in each iteration, you can only expect relatively small increases, sometimes as little as between 5 and 10%, which means that you have to do more iterations, which ultimately, of course, can add up and mean you get a significant effect. So here is, of course, being able to do this, you have to be really efficient and accurate, but ultimately we always reach cumulative results. So that's all for me. I give my word again to Helena.
Okay. So now maybe we go a little bit more detailedly into adaptive laboratory evolution. Gregor really already mentioned a few words about ALE, or adaptive laboratory evolution. This is actually a strategy that we are using often when we are relying on microbes' natural adaptability and natural selection. As cells grow and divide, then spontaneous mutations occur, and those strains that have some kind of fitness advantage in these conditions then become dominant in the population. So this already happens naturally, but we can also speed up this with addition of mutagenesis at the beginning, and you already introduce mutagenized culture into the ale, and you don't have to wait for the mutations to happen. You have already them present in the start. So you usually start with the starting strain that has some desired trait that is low. It can be growth rate or maybe resistant to something. And then you do subcultivations under relevant conditions. What is really important is the relevant conditions. They have to be set up in a way that you select the right strain at the end. So traditionally, these subcultivations were done in shake flask or falcon tubes, and you subcultivated the strains by hand, which can be laborally intensive because it is ideally to subcultivate the strains in the exponential phase all the time. You don't want the strains to go into the stationary phase, because then you select for unwanted mutations like the adaptation to stationary phase or dormancy or something like that. So ensuring that you have the strain in the stationary phase when you subcultivate it can be really labor intensive and sometimes even impossible, because the ale experiments often last really a long time. You need usually at least 100 generations to observe some kind of progress. So here in Acies Bio, we developed our own micro-ale systems that have automated subcultivations, and they are miniaturized, which means we are working in a few milliliters these subcultivations. So we have several of these systems available, which means they allow us parallel execution and they support OD-based and time-based subcultivations. So these traits that we are changing, they can be growth rates, substrate utilization, it can be product inhibition, metabolic inhibitors, maybe changing temperature that the strain grows in, or even plasmid removal, or sometimes we used our ale system also for testing of strain stability. So in ale, in every case, you start with a strain that has a low desired trait and you end up with evolved strain. These changes don't happen linearly. It usually have a few steps in between. So our micro-ale system allows us to be run in a few different modes. Maybe the mostly used mode that we have is this adaptation to production conditions. These are actually classical subcultivation, but they are automated. And we can set up a starting and finishing ODs so that the cycles can be really maintained at the same ODs all the time. And then you look at the progress of these cycles. Ideally, they would come more together with evolution or with time. And after some time, you decide to screen these strains at the larger scale. So in theory, these ale experiments could last forever, but in a real case scenario, you decide that then when you see some kind of progress, you stop the ale and you screen if there is some progress. The second really often used mode is this stressor adaptation mode, when you increase the stressor concentration in each subcultivation step. This is also done automatically in our systems, and you set up the concentration that it is increased in each of these ale cycles. So in this case, you screen also for the length of the cycles, so how much time it passes between one passage to another. And if you see that this time is getting too long, then the strain is really struggling, and maybe you need to decrease the increasing steps a little bit, or you have to keep the strain on the same concentration for some time. The little bit less used modes that we have are turbidostat or chemostat modes, where you have a constant OD or constant flow rate. But it can be useful if you have some unstable chemical or unstable stressor, or maybe if you have a continuous process. And the fourth mode or fifth mode that we have is temperature adaptation mode, where we increase the temperature in each cycle as we subcultivate the strains. And maybe a little bit about the showcases that we have. So one was the classical subcultivation ale. Here we had a strain, but with unknown metabolic deficiency and had really reduced growth rate compared to the parent strain. We didn't really know what was wrong, but we used industrially relevant medium, and we wanted to improve the growth of the strain. To speed up the ale process, we introduced the mutagenesis at the beginning of the ale, and then we started the subcultivations. So we screened the strain even after 13 days of subcultivations in ale, and also after 23 days, and significant improvement in growth rate was observed even after 30 days and also after 23 days. And the last of the showcases is a case when we had a stressor present. We had a chassis strain designed for production of some chemical, but the strain struggled with the inhibitory effect of the product. So also in this case, mutagenesis was done before each ale round. In this case, we had 2 ale rounds. We usually don't want an ale around to run for a really long time. We usually have an ale around for, I don't know, 4 or 5 weeks, and then see the progress, and then do a new round, maybe introduce a new mutagenesis and start. You don't have to start from the beginning, but from already partially evolved strain, and you get higher. So in this case, we had 2 ale rounds. With the first, we got to some kind of plateau, and we couldn't increase the concentration of the stressor anymore. And when we tested this strain on the shape scale, we saw that it grew better than the parent strain in the presence of the stressor, but the growth was not ideal yet. So there was a second round of ale with the same stressor. And as you see here on the chart, the first steps of increasing the stressor concentration are actually really fast because the strain is not really struggling. But as the ale progresses and the concentration is getting higher, the strain is starting to struggle. So we decrease these increasing steps a little bit. But after certain time, this times between passages was getting really long, and here in this case the growth was really stalled. So we even reduced the stressor concentration a little bit, and we kept the stressor constant until the growth of the strain picked up again, and then we started increase the concentrations of the stressor again. So the growth at the end of the second ale round was much better on the shaker scale. You see that it was more than 50% of what it was on the medium without the stressor, but it's not ideal yet. So additional ale rounds would be beneficial here. But already in 8 weeks, we got significant progress. So this is all from me.
Okay, thanks, Helena. Thanks, Dusan and Gregor for the insights. I think we had a lot of different use cases, and I believe that each of you had in mind, where parts of this strain optimization that we do could be used during your strain development or technology development for your products. But maybe to summarize also regarding the uses, the development and improvement of these industrial traits using strain optimization can be used for development of industrial traits in strains for diverse industrial segments, and many times there is a big time saving, especially when we are talking about multi-trait optimizations or even traits that are multigenic, so having different causative mutations, then you can maybe tackle that through this strain optimization efforts. And then finally, using that, of course, is improving the competitiveness and also eco-efficiency of fermentation technologies through higher yields and more efficient production. So that's the goal of all of the technologies that we are developing and are also competing on the market with existing technologies or other ways of productions. So we've come to the end of today's webinar. There is a Q&A section that is also scheduled now, but please also scan the QR code here and give us some opinion on what you liked, what you maybe didn't like, so that we can improve for the next episodes. I'm gonna just switch to the Q&A now. And we can start with the first question. We have some time, so I think we can probably answer majority of them. So the first question was, how long does it take you from screening to validation? Maybe Dusan, you can take over that question.
Yeah. Sure. So, of course, every project starts with a setup where we need to set up all our methods, including analytics, cultivation techniques. So we always have, let's say, a month usually reserved just to set this up. But as we begin our improvement, let's say that just to define at least one round, because we never know how many we will do based on the success. So one round, I would say, working with fast strains like Bacillus, it can be as short as, going from deep wells, retesting, shaker, reactor. I think up to 3 months is realistic. If, of course, this is a relatively short process. If you're working with a very slow strains, talking about, actinos or something like that, when just one experiment can take like 3-4 weeks, then of course this can be even double that. So I think it's quite strain dependent.
Okay, thanks. What is the throughput you can have on HPLC and MS for high throughput analytics?
So I think again here is the method duration is here the main factor. So the methods can be as short as 3 minutes, but they can be as long as 20 plus. Again, if you look at the profiles of metabolites or even try to detect some impurities, this usually means very long run times, half hour plus, and then you can do the math. Per day, you can test with a good method several hundred, with a more complicated method, less than that. So, but I say that usually the microbiological work is basically continuing while you're waiting for the results of the first set of samples. You're already finishing the last one. So our analytics team has quite some time to finish their analysis.
Maybe just to add that in our analytical department, we have more than 30 HPLC instruments and almost 10 of the MS-based instruments. So if necessary, we can really increase the throughput by including more instruments in one project.
Okay. Okay, which scale for ALE is representative, and is it possible to perform ALE for filamentous organisms?
Yeah, filamentous organisms are really harder to work with in ALE. Yeah, because we have smaller volumes. We are working actually right now on that with different kinds of vials to be used. So you don't get this wall growth or things like that. But currently we are not working with filamentous organisms in ALE usually. It has to be a more unicellular growth.
So it is work with filamentous organisms, of course, is different in many ways from working with I would say classical bacteria. You have to take into account the physiology of the growth, but also that if you have a mutant, then you need to usually go through some sort of single cellular form like a spore, which then takes you to the next generation. Yeah, so the methods have to be adapted, of course, and as Dusan mentioned, then it also takes a little bit more time when you work with such organisms.
Okay. How many candidates do you take from each evolution iteration stage to the next one?
Here, again, depending on where you're starting. So if you're starting with a very low yielding strain, this is the, I would say, easy part where you're just kind of picking the low hanging fruit. And usually you don't need to test many because some will really significantly jump out. When you go to higher titers, then again, the differences are smaller, which means you have to take more to then, statistically include even the ones that are on their bottom part of the error. So would say this really depends on the title where you're starting, but these are usually just a few % of the initial number of the mutants where you started.
Okay. Can you run experiments in multi-phase different layers condition in your ALE?
I'm not sure exactly what this means. Like all phase, yes, this was already done in our systems. It disturbs the OD readings a little bit, but it can be managed. Yeah, we had already some projects that were run on oily phase.
So like if you have an overlay, for example, then that was perfect. Okay. On slide 14, you show the graph with relative yields of 112 clones. Which organism was that?
Yeah, so this was a Bacillus subtilis experiment.
And probably shows also the variability of the genetics, right?
Exactly.
Okay, for automated ALE, the monitoring is made by measuring the microorganism growth. What if the target is not a secreted product?
Yeah, the conditions in ALE have to be set up in a way that you are screening for the right traits. So yeah, ideally, if the growth is coupled with the production, then you can also screen for the growth, or you have to have the conditions somehow to be stringent enough to not to lose productivity. Maybe some inhibitors, or to have really short ALE rounds to not to lose the productivity.
Mm-hmm. Thanks. Okay, so for all of you who didn't catch the QR code before, here is again for getting the feedback for the webinar. And we do have another question here in the chat. So do we get reliable data using high throughput screening using filamentous bacteria?
Yeah, I would say so, definitely. So we, in our early days, we were working mostly with actinomyces, which are filamentous bacteria. Of course, it's quite tricky to get them to, when you do mutagenesis, you have to ensure that this is a single cell clone, so one genome per cell. This can be tricky, but there are ways to achieve this. So usually it's either going through spores, or in the most difficult, let's say, case, I would say you have to work with protoplasts. This is very difficult, very touchy work, but it can be done.
Mm-hmm. Okay. So if there is any questions, you can still raise your hand and we can answer it. Otherwise, As you can see on my camera, we have really nice weather outside, and we will conclude with that. Oh, there is a question from Maria appearing. You can also talk. Okay, thank you. Actually, another question, but we really value the feedback. I hope it was insightful. I will let you think about, what are the aspects where you would also need a strain optimization, yeah. We are looking to get a feedback from you. Maybe you can contact us if you need any of our services. But otherwise, we will keep you posted with the news from Acies Bio and when our episode 2 will be scheduled. Have a nice day and bye-bye.
Thanks, bye.
Bye.